Good morning to my dear students. In our last class, we have discussed the detailed account about the structure of striated muscle. In our today's class, we are going to discuss about the contractile protein, that is actin and myosin, which forms the light and the dark bands of the muscle fiber. So let us discuss the actin first. The figure A is showing you the actin, a figure of the actin diagram. Now every actin is made up of two F actin. F here stands for filamentous. So every actin is made up of two filamentous actin which is helically coiled to each other. You can see here the green color beads are there, and these two strands of these green color beads are there, and they are helically coiling. They are showing you the coiling like the DNA is showing the coiling. So the two green stripes are there which are coiling helically around each other. Each F actin is a polymer of G actin. Now here G stands for globular. So the every green bead is the G actin, and when the number of G actins are arranged, then it becomes filamentous actin. So every filamentous actin is a polymer of G actin. You very well know what is a polymer. Polymers are the long chain compound where the monomeric unit is repeated again. So for filamentous actin, the G actin unit is getting repeated. Okay. So two filament of another protein, tropomyosin, are also running close to F actin. You can see here the brown color strand. This brown color strand is another protein, that is tropomyosin, and this tropomyosin is running around the F actin. Now on the tropomyosin you will find a complex protein, that is troponin, and this troponin is distributed on regular intervals on the tropomyosin. In the resting state, the subunit of troponin masks the binding site of myosin on the actin filament. So here you can see the three small beads that are located on the tropomyosin at regular interval. This is known as troponin, and this troponin is point where the head of the another protein, that is myosin, is going to bind. So what this troponin is doing? This troponin is covering the binding site of myosin head. Now just below there is a B structure which is giving you the account about the myosin. Now this is the single unit of myosin, that is a monomeric unit. So let us discuss the myosin. It is a polymerized protein and is formed of monomeric unit, which is shown here in the figure. It has two parts, globular head and a tail, known as heavy meromyosin and light meromyosin respectively. The head is called heavy meromyosin and is represented by HMM, and tail is represented by LMM, that is light meromyosin. Heavy meromyosin is coming outward at regular interval. The head has the active site for ATP enzyme. You can clearly see the head is having the actin binding site also, and it is having the ATP binding site also, right? So this is the complete structure of the second protein, that is the myosin. Now this actin binding site is going to stuck on the part of the troponin. The troponin is covering the myosin binding site. So what will happen? The head of the myosin is going to bind to the site, the troponin will be removed, and this head will bind to the tropomyosin. So these are the two structures of actin and myosin. They are very important. Please read them carefully, and if you have any doubt, you can contact.